The next presenter is Jonah Sinick from Equator Therapeutics. There's Jonah. Hi, Jonah.
Hi, good to see you. Thanks to everyone for the opportunity to be here today. So let's see. Are my slides visible? Okay, great. So yeah, I'm here today to tell you about a company that I founded a few years ago now, Equator Therapeutics. And we're developing a drug to burn fat by activating natural heat production. So when people talk about weight loss, the most salient things are dieting and exercise. So reducing caloric intake and burning those calories by exercise via where food gets converted into ATP. But there's a third essential piece of the equation, which is heat production by mitochondria, which is no less important for maintaining healthy body weight. This mechanism of natural heat production was significantly more active in ancestral times than it is today. So our ancestors lived with frequent exposure to cold, and their mitochondria were quite active in keeping them warm. Whereas nowadays, with comfortable living temperatures, we're no longer burning as much fat in this way. Basically, we are looking to restore natural heat production to levels more representative of what our ancestors enjoyed, thereby burning fat. So this approach, there's a sense in which our approach is not at all new. Compounds that activate heat production in humans and burn calories have been known for 100 years. So the most famous of these compounds is DNP, 2,4-dinitrophenyl. So this was a compound used in explosives during World War I. It was found that factory workers who ingested the dust lost weight. So within 10 years, DNP was being used as a weight loss drug. It was used by 100,000 Americans. It was highly effective. It was standard for people to lose 3 pounds a week. So really kind of phenomenal efficacy, but it ultimately proved to be unsafe and was banned by the FDA. But ever since this discovery was made, there's been this dream. What if there was a way to do this safely? So yeah, there's been a lot of follow-up research on this. Here I'm showing you data from a paper by Dr. Gerald Schulman's lab, who you heard from earlier this week. So they studied a controlled release version of this toxic molecule from the 1920s, DNP, finding phenomenal effect sizes for reducing blood glucose, reducing insulin, and reducing liver triglycerides. So this is a solution. This approach is potentially a solution not only for obesity but for metabolic disorders holistically. And yeah, now why I made reference to DNP being the heat production being artificial in nature. So just for context, natural heat production occurs when you have the Krebs cycle and electron transport chain converting nutrients and oxygen into voltage, which powers currents through the inner mitochondrial membrane that pass through a thermogenic protein to produce heat. So some of you might, some of you are familiar with brown fat, where you have lots of this thermogenic protein UCP1 producing heat. That's what this is referring to. So in contrast with natural heat production, you have heat production induced by DNP and similar compounds, where you have the currents flowing through the membrane directly without being mediated by a transporter protein. So these compounds like DNP are called protonophores, and they induce currents not only in the inner mitochondrial membrane, but also in all other membranes. Why is this not good? Because so the specialized heat production mechanism of mitochondria co-evolved with ATP production to be compatible with ATP production. If you're inducing currents directly through the membrane, the mechanism is not necessarily compatible with ATP production, so you can have ATP depletion. You also have the currents in other membranes, like the plasma membrane, disrupting electrical signaling in neurons and cardiomyocytes. And you have disruption of proton gradients in endosomes and lysosomes, which are crucial for autophagy. So DNP and other protonophores, they have a pro-aging effect by interfering with autophagy. So in contrast with that, the natural heat production mechanism, the heat is produced by currents going through this transporter protein. The transporter protein physiologically is activated by long-chain fatty acids. And this mechanism was characterized by my co-founder, Yuriy Kirichok, in his lab at UCSF, first in brown fat in this paper in Cell 2012, then in all other tissues in 2019. They characterized this mechanism of long-chain fatty acid-induced heat production. Now you might wonder, well, why not just give people long-chain fatty acids to activate this heat production? But unfortunately, if you ingest long-chain fatty acids, they don't reach mitochondria intact. So they're not a viable drug candidate. And in parallel with that, before our work, long-chain fatty acids were the only known activators of natural heat production. So people were using protonophores and were stuck in that way. So, sorry, just a moment. I seem to have missed a slide. Okay, yes. So at Equator, we have finally identified the first selective activators of natural heat production. On one hand, we've greatly expanded on the universe of known compounds to activate heat production. So, between the discovery of DNP in World War I until quite recently, there were only 25 known molecules to activate this mechanism. And since we incorporated at Equator, we identified 500 compounds that activate heat production, which crucially include the first compounds that do so through the native pathways, in contrast with these protonophores that induce currents through all membranes. So here we have 4 lead series. We are doing medicinal chemistry optimization on these series. And what's made it possible to make progress on this 100-year-old problem is this unique method developed by my co-founder, Yuriy Kirichok, to directly record the currents through the intermitochondrial membrane, which involves isolating individual mitochondria from cells, teasing out their inner membrane, and then applying electrophysiology to the membrane. And so this is the method that allowed him to characterize the mechanisms, and this is also the method that allows us to do target engagement validation, which is something that otherwise is not possible. So just to give you a sense for the sort of data that comes from this method, here is a comparison. The first row is long-chain fatty acids, the native activators of heat production. The second is DNP, the toxic molecule from a long time ago. The third column is our compound. One of them. We have many such compounds, and then in the top row is the current in the inner mitochondrial membrane. The bottom one is in the plasma membrane. So for long-chain fatty acids, the natural activators, you have current in the inner mitochondrial membrane and none in the plasma membrane. That's what we want to see. With DNP, you have occurrence in the inner mitochondrial membrane, but also in other membranes, which we don't want to see. And then here is a representative graph of one of our compounds where you have currents of the same magnitude as DNP in the inner mitochondrial membrane, but none in the plasma membrane. So we have these entirely new classes of activators of mitochondrial heat production, which we know to burn calories in cells. And we are in the process of optimizing these compounds for bioavailability to be systemically available drugs to treat obesity and metabolic disorders. I've been fortunate to have really great collaborators. I mentioned my co-founder Yuriy at UCSF, who developed the underlying technology. We are working closely with Catherine Widdowson, formerly at GSK, distinguished chemist, and Lilia is our bench scientist who generated the data that I've showed you today. Rebecca is our project manager who's done a great job with helping Catherine and us interface with CROs. And yeah, we have excellent expertise on the metabolic side. Jerry Schulman recently joined our scientific advisory board alongside with Andre Duneif and Michael Grade. We are currently, so we've raised 2.5 million to date from Y Combinator, Assorted Angels, 100 plus capital, and most recently received a phase 1 SBIR grant from the NIH. We are currently raising a round of $5 to $10 million to convert these lead molecules that we have into drug candidates, ultimately to select a final drug candidate for IND enabling studies, and which will be followed by a Series A to raise for human clinical trials, leading to a liquidity event, IPO, partnership, or acquisition. So yeah, just to summarize, mitochondrial heat production is a powerful fat burning mechanism, can reverse metabolic disorders. And in the 20th century, my co-founder Yuriy likes to say that we had this antiquated technology of the 20th century, these non-specific protonophores, which could induce heat production, but in a harsh and unnatural way. Yuriy's application of mitochondrial electrophysiology made it possible to develop the first selective activators of this natural heat production mechanism that's crucial to weight regulation. And Equator has multiple lead series of such compounds now. And if we hit all of our technical milestones, we will have a drug that can completely reverse metabolic syndrome safely, thereby addressing the subject of the conference today. So yeah, thanks again very much for your attention and interest.
Thanks, Jonah. Yes, 100 plus capital is invested in this company. I love the fact that there's a real world proof of concept that this mechanism works, and now you're just trying to make it safer. And so there's a couple of questions from the audience, one from Thomas actually. So I'm just going to start with those ones. The first one is this going to make people sweat? Personally, I am cold all the time, so I would like to be a little bit warmer. But will it make people hot if they're taking this drug, and will it complicate menopause?
Yeah. So when it comes to whether it makes people hot, we're targeting increasing heat production by about 25%, which is still it's much lower than the heat produced by walking. So the drug at therapeutic doses will not make people hot. Up to a point, your body deals with excess heat by dilating the capillaries, increasing the surface area of the outside world against your blood, reducing heat that way. So yeah, the therapeutic levels, you'll be at body temperature.
So you wouldn't notice it. You'd be taking it and burning more heat, but it wouldn't be super noticeable.
Yes, that's correct.
Right. And any comment on the menopause question?
Yeah, so I guess Thomas, if you could elaborate on what you have in mind. I mean, I know sometimes people talk about heat flashes with menopause. Is that?
Yeah, it was just the same variant. If you're telling me that it's a fraction of walking in terms of heat production, then maybe menopausal women will blame me, but maybe that's not a big issue.
Okay. All right. Well, then let's move on to the next question from Michael Zemmel. He says, interesting approach. What is the evidence that it will have an impact on cardiometabolic outcomes of obesity? To do that, these molecules would need to have a greater effect on reducing visceral versus subcutaneous fat.
Right. So I guess visceral fat burns off before subcutaneous fat in general. So as you burn fat, first you burn through the visceral fat, then you burn subcutaneous fat. And it's even the case that you can burn visceral fat without reversing obesity, which is an approach in this field to actually just make a liver targeted compound, which burns the fat off of the liver and then prevents VLDL from being secreted. And VLDL is what's responsible for maintaining the visceral fat deposits. So you can actually just go a liver targeted route and selectively get rid of visceral fat, but we are hoping to burn both visceral fat and subcutaneous fat, returning people to normal body weight.
Got it. Yep. And so one last question. What surprised you the most in your journey going through, finding this compound and working towards, bringing it to trial?
Yeah, that's a good question. I think for me, the main, what I've found most exciting about this is the precision of the assays that Yuriy and his colleagues have developed. Just how robust and replicable the data is from these assays, because I've worked with data from many different kinds of biological assays, and just with these physical measurements, I've never seen something so precise. So that was really eye-opening to me.
Great. Well, thanks, Jonah. I think we'll move on to the next presentation. Thanks for telling us all about Equator.
Yeah, thanks very much to you as well.